Hello everyone, my name is Chihiro Tsunoda. Today I'm going to talk about a new implementation of how cyclodextrin works in the presence of multiple drugs that interact with each other. This is a title of this presentation. In recent years, polypharmacy in which several drugs are prescribed simultaneously have become common among the elderly and other patients suffering from a variety of diseases. However, it has been reported that the use of multiple drugs in combination with each other can decrease the efficacy of the drugs. In fact, we have previously reported that diclofenac, a type of NSAIDs, form the ion pair complexes with famotidine, a type of H2 receptor antagonist, and that the solubility of diclofenac is reduced when it coexists famotidine. Long-term administration of NSAIDs is well known to cause peptic ulcers, and the proton pump inhibitor famotidine is the most commonly prescribed drug for the treatment of peptic ulcers. In this context, such combination of acidic and basic drug is considered a polypharmacy event that can occur in daily life. In this study, we focused on how cyclodextrin works against the unexpected decrease in drug efficacy caused by the coexistence of multiple drugs. Cyclodextrins are known to improve by encapsulating hydrophobic drugs, but most of these reports are on the effect of cyclodextrin on a single drug, and there are few reports on the effect of cyclodextrin in the presence of multiple drugs. So, in this study, how do they function in the presence of multiple drugs that may interact with each other? This is a subject of this study. So, in this study, we aim to clarify the effect of cyclodextrin complex formation on the interaction between acidic NSAIDs and basic H2 blockers, which are already known to interact with each other. Indomethacin and diclofenac were used as a model of acidic drugs. Cimetidine and famotidine, imidazole, histidine, arginine, and caffeine were used as a model of basic drugs. First, we prepare phase solubility diagram to determine what ratio of drug and CYD form complexes in solution and how the solubility of the complexes changes. In this study, beta-cyclodextrin, which is composed of 7 glucosides, were used among the many CYD family members. The complex tension efficiency, CE, which is an indicator of the affinity between the drug and CYD, was calculated from the slope of the phase solubility diagram. The addition of basic drug to an acidic drug was performed using two different methods, co-dissolved model and pre-dissolved model. Co-dissolved model assumes that basic drug and an acidic drug are taken at the same time, and the basic drug is added in a solid state. While pre-dissolved model assumes acidic drug is taken after the basic drug has taken before. In pre-dissolved model, the basic drug was added to the solution after dissolving the basic drug in phosphate buffer. In phase solubility diagram, the solid line indicates that the drug was added in a solid state, co-dissolved model, and the dashed line indicates that the drug was added in a dissolved state, pre-dissolved model. The following are the experimental results. First, the solubility of indomethacin was greatly improved in the presence of cimetidine, imidazole, arginine, and caffeine. The CE of the indomethacin CYD complex was significantly increased by the addition of imidazole and arginine by co-dissolved model and caffeine by pre-dissolved model, respectively, from the state where nothing was added. In the diclofenac, the solubility of diclofenac was greatly enhanced in the presence of cimetidine, imidazole, arginine, and caffeine. The complex tension efficiency, CE, was 8-fold higher with arginine from the state where nothing was added. These results indicate that the addition of imidazole ring containing compounds to indomethacin strongly enhanced the solubility and affinity of the indomethacin-CYD complex, while the addition of arginine with guanylyl group to diclofenac strongly enhanced the affinity of the diclofenac-CYD complex, even for the same acidic drug. This suggests that basic drugs' enhancement of affinity and solubility of the acidic drug CYD complex is structure dependent on the acidic drug. Next, we move on to the basic drugs. These are result of phase solubility diagram of famotidine. The solubility of famotidine at low concentration of CYD at 48 hours of shaking was significantly decreased in the presence of diclofenac and indomethacin, while at high concentration of CYD, the decrease in the solubility due to diclofenac and indomethacin was improved. On the other hand, there was no significant difference in the solubility after 120 hours of shaking in the presence of indomethacin and diclofenac. The figure shows that intermolecular interaction points between drugs based on NMR spectra. The radius of the circle indicates the magnitude of the change in the chemical shift for each carbon. Previously, we have reported from this carbon NMR measurement that diclofenac and famotidine, when in coexistence, shift the amino group of diclofenac and the guanylyl group of famotidine to higher magnetic field, resulting in a stacking interaction from above the aromatic ring. Improvement of solubility of famotidine even in the presence of diclofenac at the high concentration of CYD suggests that high concentration of CYD improves the dissolution rate of each drug by separating the interaction between famotidine and diclofenac. On the contrary, decrease in the dissolution rate of famotidine at low concentration of CYD indicates the difference in the concentration-dependent function of CYD on the diclofenac-famotidine interaction. Cimetidine was not affected by any solubilizing effect of CYD in the presence or absence of acidic drugs. Also, here are the dissolution profile of indomethacin, diclofenac, famotidine, and cimetidine, respectively. From these results, you can see how the solubility of each drug changes with each acidic and basic drug interaction. Here is a summary of this study. CYD increases the solubility of acidic drugs synergistically and structure-dependently with basic drugs containing imidazole ring and guanylyl rings. The decrease in solubility of famotidine due to static interaction between diclofenac and famotidine can be improved by the addition of high concentration of CYD. This indicates that CYD can be used to improve physicochemical properties of drugs, even under the administration of drug combinations that interact with each other. Thank you for listening.